Good morning, everyone, and welcome to Atara Bio presentation at the Alliance for Regenerative Medicine Cell and Gene meeting on the Mediterranean 2021. Unfortunately, still a virtual meeting there. After the forward-looking statement, I would like to highlight the mission of Atara Bio, as our mission is so important for us in everything we do every day, which is really to transform the lives of patients with serious disease through pioneering science, teamwork, and a commitment to excellence. On this slide, you see what we call our champions, which are patients affected by this serious disease that are really fighting every day to address the disease and to cure, if possible, with the appropriate treatment. And we're here to help them in that fight. Now, Atara is truly a unique company in the sense that we are not only a leading allogeneic T-cell immunotherapy company, but we have 5 key elements that are truly unique to Atara. First, we have a differentiated allogeneic cell therapy platform. We have a scalable EBV T-cell platform and technologies enabling us to develop multiple allogeneic cell therapy. Second, we have with Tapcell a true first-in-kind, a late-stage oncology program that could be very well the first ever allogeneic T-cell therapy to be approved in the US and in Europe. We are planning to complete our BLA in Q3 2021 and to file a market authorization application with the EMA in Q4 2021. And all this is underpinned by very strong clinical data. We have with AT188, our second clinical stage product, a potentially transformative MS treatment, which is right now already in randomized controlled trial. And we're working hard on this study with placebo-controlled data expected within 18 months to enable then pivotal studies and partnering opportunities. We have also a very unique next-gen allogeneic CAR-T portfolio, recently validated by a strategic collaboration with Bayer on mesothelin-targeted CAR-T. And this is really a number of programs that we believe are truly competitive as they are designed to address the current limitations of autologous and allogeneic CAR-T. And all this portfolio of transformative therapies is being supported by proven technical capabilities in terms of advanced process science, as well as our own pre-commercial manufacturing capabilities there. What we do at Atara is really to leverage EBV T cells coming from healthy donors. And we do so in addressing either EBV-associated disease or other type of hematological and solid tumors through manufacturing and engineering the cells. First step is to make product that can address at the core the root cause of EBV-driven disease. And here we have Tapcell and AT188 that are being manufactured from healthy donor cells to really be very specific and selective to address EBV-driven cancer or autoimmune disease. We have already demonstrated a very favorable safety profile, encouraging efficacy, and we have a very robust manufacturing process with, in that case, no genetic engineering of these cells. What we do as well is to go one step further with these EBV T cells coming from healthy donors, where using next-gen CAR technologies, we can engineer next-gen CAR-T or TCR-T as allogeneic CAR-T or allogeneic TCR-T. This allows to retain the beneficial features of EBV T cell from healthy donors, but then to have a CAR-targeted activity allowing durability of response and persistence of the cells. And these cells could be further modified to add other features to make them more armored, especially to address specific challenges of solid tumors. Now, our technology, our platform, we believe has the possibility to transcend the limitation of current cell therapy by truly harnessing the power of these EBV T cells. In terms of durable efficacy, we've already demonstrated this to be the case with this type of allogeneic T cells. We've also demonstrated favorable safety profile in many patients with low level of GVHD and the ability in preserving the natural states of these cells with an adapted phenotype to truly have persistence of the cells enough for durable remission. The platform we are leveraging here is already scaled up with flexible off-the-shelf type of cells that are in our inventories. And we have proven our ability to deliver within 3 days from inventory an allogeneic T-cell therapy to patients. So from a product supply and also patient access and experience, where we have truly developed off-the-shelf convenient cells that can really satisfy patient needs, we believe that we have some significant differentiation and benefits compared with other technologies. Now, our portfolio is made of a very robust T-cell immunotherapy pipeline, not only with Tapcell in several indications, but also ATA188 in progressive multiple sclerosis, and a number of CAR-T programs that are approaching the clinical phase now. I would like to spend some minutes on Tapcell, as this is really a unique therapy that is linked with the fact that EBV, which is a very common virus, 95% of the world population is infected by EBV, can cause very rare and serious cancers in patients with impaired immune function. And Tapcell has indeed the potential to help patients, thousands of patients every year, to address their disease progression and to be able to respond to the treatment and have a longer life expectancy. We have already a phase 3 study called the ALLELE study in previously treated EBV positive PTLD. And we have started recently a phase 2 multi-cohort study, having with Tapcell a product again that specifically targets and kills EBV infected cells, addressing the disease at the source. This product, based on our current data from phase 2 program, as well as a recent interim analysis of our phase 3 study, as we believe a compelling value proposition for EBV positive PTLD and healthcare system, patients and healthcare system, having not only a higher durable treatment effect with up to 50% and higher of objective response rate, and also a survival at 2 years that is over 80% in patients for which there is no approved treatment today. But this is combined with an excellent safety profile with no CRS or neurotoxicity and no PTLD-related mortality, and a low burden of administration, as you don't need to do any pre-treatment for Tapcell use, and it's just a 5 to 10 minutes IV infusion with a 2 hours monitoring. And all this is truly off the shelf, being delivered in about 3 days with T-cell from our own inventory there. So truly a compelling value propositions in its first indication of PTLD after prior therapy. Now we're also developing actively through a phase 2 study up to 6 additional indications for Tapcell in population for which we have already clinical data showing some interesting level of efficacy and safety. And we presented recently new data last year at ESMO and then at ASH, showing this high level of efficacy and acceptable safety in this type of patients. But pursuing that studies allow us really to be able to expand the label of Tapcell beyond its first indication, therefore to address the needs of many more patients and to have an even larger business potential for this product. Moving now to ATA188, this is truly a potentially transformative MS treatment, as the characteristic of the product of ATA188 as an allogeneic T-cell therapy targeting specific antigens of EBV that are always present in MS patients allows to address the disease at the core. We've already seen an acceptable safety profile in our phase 1A, which is very important for this type of patients. We've also seen very excitingly sustained disability improvement in our phase 1A, especially at high dose. So that shows the potential of this product to halt or reverse disability progression, which is a true unmet medical need in progressive multiple sclerosis. The product is able to penetrate the CNS, as we've seen that EBV-specific T cells can traffic and cross the blood-brain barrier. And it's truly a precision targeting that limits off-target activity, because we are just eliminating the B cells that are infected with EBV, and that we believe are at the core of the disease progression there, therefore addressing the underlying biology of the disease. Now, we've presented a few months ago at the Charcot conference in November, very encouraging data, especially in high dose cohort 3 and cohort 4 of our phase 1A study, where we've seen in progressive MS patients up to 50% sustained disability improvement. We mean by that disability improvement confirmed at 2 consecutive time points in the highest dose cohort at 15 months. And this is supported by improvement in patient-reported outcomes. So very encouraging data there that led us to accelerate and update a phase 2 study, a randomized control study, for which we have had a good alignment with the FDA. Now we have a very strong, robust phase 2 program that is targeting an enrollment of 80 patients with progressive MS. And this randomized control study, for which we have started to enroll patients last year, will lead us to have an interim analysis in the first half of 2022, with exciting data that will allow us then to adapt our development program in discussion with the FDA. The collaboration with Bayer now is moving ahead for mesothelin-targeted CAR-T in various solid tumors, including, of course, advanced mesothelioma, but also lung cancer and possibly other tumors overexpressing mesothelin, like ovarian cancer and pancreatic cancer. We have here built a CAR-T with our collaborators at Memorial that is very unique, very differentiated, having not only a binder that ensures the right level of targeting and safety for essential tissues, but also a novel costimulatory domain and a PD-1 dominant negative receptors, both features enabling us on one hand to ensure more functional persistence of this CAR-T in the tumor, but also the ability to resist the immunosuppressive environment for this PD-1 dominant negative receptor that allows to have an intrinsic checkpoint blockade built in within the CAR-T. This product called ATA2271 is the autologous version. It's already in the clinic, and we have enrolled several patients now with our collaborators at Memorial. Meanwhile, we are accelerating the development with Bayer of ATA3271, which is the allogeneic EBV mesothelin CAR-T version. And this product has benefited from very exciting preclinical data, which we presented at SITC last year, showing potent anti-tumor activity without alloreactivity and functional persistence there. We expect the IND for that very exciting allogeneic CAR-T to be submitted in Q2, Q3 2022. So as a conclusion, we have 3 strategic priorities at Atara that are driving long-term value for the patients, addressing unmet medical needs in a transformative way, as well as for the company. With Tapcell, the first-in-kind allogeneic T-cell therapy that is preparing for historic regulatory filing as the first ever allogeneic T-cell therapy to be filed with the FDA and EMA. With ATA188, which is a potentially transformative MS treatment that is already in randomized controlled trial versus placebo. And of course, with our next-gen CAR-T program, where we have several competitive programs now close to start in the clinic. If you look at the next 18 to 24 months, we are planning to make rapid progress on multiple fronts. One hand, on Tapcell, we hope that within the next 18 to 24 months, we have the potential for the first ever allogeneic T-cell therapy to be approved by the EMA and the FDA. And then on ATA188, we should have disability improvement data from a randomized controlled trial with the potential to unlock multi-billion-dollar opportunity in this autoimmune disease that is affecting so many patients across the world. With the CAR-T, we'll have clinical data on both liquid and solid tumors over the next 18 to 24 months to validate the approach we are taking with these very unique and differentiated allogeneic CAR-T platforms, and all that being supported by our manufacturing platform with a commercially scaled up and validated allogeneic T-cell therapy platform there. So as you can see, making huge progress at Atara for the benefit of patients and to create significant value inflection point for the company. I would like to end in thanking you for your attention and wishing you a great conference.